first up today, we have our very first speaker, and probably the timing on the video. I just wanted to kick things off with the Kicking Cancers KRAS, but we've got online with us today. I am honored to present to you our first speaker, and he comes to us. His name is Darryl McConnell. He's a Ph.D. and he's at Boehringer-Ingelheim, which I've been practicing saying for quite some time now. And they are also one of our sponsors, and I am thrilled to present him to you guys today. I specifically asked if he would come and do a presentation to us, because what he's got is going to kind of break it down as far as what is a KRAS, what does it do, how did it happen, and what does it mean in terms that we can understand, but at the same time have a sense of scientific balance. Darryl McConnell is currently the Senior Vice President and Research Site Head at Boehringer-Ingelheim in Vienna, Austria. And his goal is to discover new chemical therapeutics for cancer's big four and beyond with the team at BI. Okay, Darryl commenced his career with Boehringer-Ingelheim in 2002 as a research lab head and was currently the current role since 2015. His interests outside of pond building includes drugging protein interactions, kinases, and pushing the frontiers of proteins for cancer patients, which I hope, Darryl, you're going to explain to us a little bit more. Fragment screening, protein, cystology, and I think I'm going to stop there because this is over my head. So thank you so much for joining us today, Darryl, and I appreciate your being here. So I'm going to leave it to you to do a little explaining of what a KRAS is. Thank you.
Terry, thank you so much. I'll just share my screen here, get the technical stuff going. So yeah, thanks guys. It's a real honor for me to speak to you. I've been looking forward to this so much. So thanks. Let me just bring you down here. Yeah, thanks so much. I think patients, caregivers, physicians, researchers, I think all do a fantastic job, and I know we're all struggling with things and not giving up on all fronts. So it's great to be part of the KRAS Kicker family, and I think it's awesome that this community is starting to grow now, because this is the decade that we're gonna drug a lot of the KRAS mutants, I think. So I have the privilege of presenting on behalf of a wonderful team at BI. That's a little tip, Terry. If you can't pronounce it, you just say BI. I'm an Australian, actually, in Austria. A chemist by training, and yeah, really looking forward to giving you an overview of KRAS. I've called the talk sort of drugging all the forms of KRAS. I want to give you a little bit of background, as Terry mentioned, but also let you know where we're at, where the field's actually at, not just the clinical aspects, which Parsi and others will do a better job informing you on than me, but also the preclinical part. So let me just get, there we go. So I'm an employee of BI. I won't be talking about off-label use today. Just some numbers. 1 in 7 of cancers are driven by KRAS about. If you look at the US, only the US, there's about 200,000 newly diagnosed patients every year with cancers that are driven by KRAS. So this really is a huge need. I don't need to mention this to any of you, of course. Actually, 1967, as Terry mentioned, it sort of started, and then 1982 was an important year where it was really identified in human cancers. This year, fantastic year, Amgen and sotorasib approved as the first G12C mutant drug. Is absolutely spectacular. About 9 different KRAS mutants make up 90%, and I'll talk about that. G12C is one of them, number 3. And KRAS has got 2 forms, an on and an off form. And there's actually 2 different, what we call splice variants of KRAS. So it's actually, although a small protein, actually a little bit complicated. So this is where it all started. It was a German guy, actually, who left to the US and was looking at viruses in rats in this case, and actually found that this it was actually a mouse leukemia causing virus. But when he put that in a rat, the virus actually stole a gene from the rat and put it back in and mutated it when it was putting it back in. And it was actually, he didn't know at the time, but that gene that the virus stole from the rat was actually KRAS, and that's why it bears his name. Werner Kirsten was his name, but it wasn't actually until 1982 that Channing Durr really identified KRAS as the gene in human cancers, and identified that it was actually the KRAS gene that mutated. And this is, of course, been the hugely frustrating thing for everyone, that it's been so long. You know, it's taken us till this year to get the first drug against KRAS. So, but I think things have changed a lot now. Technologies have changed. Some of the the things that Terry mentioned, these new approaches that we're using in drug discovery have changed the game. Now, I put this, it's a bit complicated, but I just wanted to, because I'm sure you're sort of, as patients, particularly inundated with all these technical things, and you probably know our DNA is in our chromosomes. We've got 46 chromosomes. So it's this DNA that gets really packed in, and it's built out of these codons, they're called. They're essentially sort of letters in the alphabet. And this is where KRAS is situated. It's actually on the 12th chromosome, right about here. And it's actually got, of these so-called base pairs, it's actually got a lot of them, 46,000, but it's only these little colored bits that actually get taken out and used to form KRAS. And we've heard a lot in the last years about mRNA. That's what actually happens. KRAS the gene gets turned into 2 different mRNAs, which then get what we call transcribed into proteins. And these are the 2 forms of KRAS, KRAS 4A and 4B. And they only really vary on this little, what's called the variable region. And as you probably know, well, I'm sure you know, the actual positions that these mutations happen are largely at positions 12 and 13, but also at positions 61 and positions 1 and 46. But there are, of course, other low frequency but rare mutations in KRAS as well. And this is what it really is. So Channing Durr called it the beating heart of cancer, and you can sort of see why. It actually, this is a picture of KRAS, right? So this is the protein itself. This gray sort of spaghetti thing. That's what the protein actually looks like, and you see the heart form. And it's got this GDP, GTP. This is a nucleotide. This is just a small sort of molecule, which is what this KRAS uses to switch between the on and the off form. And in normal cells, KRAS is mostly turned off. And the problem with cancers is there's this mutation. This is position 12, for example. One mutation happens and it sort of locks KRAS in this on conformation, more so in KRAS, and it actually in cancer cells, and it actually beats. Hopefully you can see this little animation. It actually beats on and off. So in normal cells, you see here when it's beating, it stays mostly in the off form, but in cancer cells, the opposite happens and it stays mostly on the on form. And that subtle difference actually has drastic consequences in that it causes cancer. And I know, biologists, I'm a chemist, so I can say this about biologists, they draw these really complex sort of schemes about how the signaling works. It's actually very much like just an electric circuit. We have cells, and outside of those cells, input is given into the cells. In this case, they're called mitogens, and these are small sort of peptides and proteins that make cells divide, and we need those, of course. And there's a volume control, and in the case of KRAS is clearly at the center of the volume control, but it's very complex setup right at the membrane of the cell, so right just under the surface of the outside of the cell. It's then got a circuit. You might have heard of MEK and ERK. This is actually what brings the signal down into the nucleus, where that mRNA I talked about before starts to get transcribed and then turned into proteins. And this is essentially how the signaling works generally, but in this case, KRAS. And it's interesting to note that in cancers, it's these so-called receptor tyrosine kinases, EGFR, you might have heard of, KRAS itself and BRAF that are mutated. And it's actually almost essentially mutually exclusive with the RASopathies. You might have heard of Noonan syndrome, and there's another disease called neurofibromatosis 1. And it's actually these sort of tuning molecules, if you like, in this case, to tune the volume of KRAS, that are actually mutated in those RASopathies. And, you know, I don't need to tell you all, this is a sort of a graph now of the different cancers with regards to those sort of around 200,000 newly diagnosed patients every year in the US. And colorectal, pancreatic, and lung cancer, we know are the three, but there are many other diseases. And one thing Sandra probably online mentioned via Twitter, that the amplification of KRAS, and I want just to highlight that here. You see in blue are patients who have mutated KRAS, but KRAS is also amplified. That means instead of just having one copy in a cell, it has maybe 10 or more copies. And you can see here there are a number of different cancers that actually have a significant amount of KRAS amplified driven tumors. I mentioned this at the beginning, KRAS has more than one target. Unfortunately, if it was all G12C, this would be fantastic, but it's actually a number. This is all cancers put together, and G12D is the most prevalent, followed by G12V and G12C. So these are amino acid changes where, in this case, a glycine to an aspartic acid. That black dot I showed you happen. And importantly, and this is often forgotten, I hope that's still working with the top there. The fourth most prevalent alteration in KRAS cancers is KRAS amplification at 8%. So I really think it's important that we come up with drugs for all of these, including a G13D, G12R, particularly difficult one, I have to say, and G12A, etc. When I hope that's okay. I hope that's not hassling at the top of the screen there. When we think about at Boehringer, when we think about how do we think the medicines for KRAS are going to pan out in the future, we think that there's going to be two classes. One, the selective inhibitors. So this is compounds as we've got now in the clinic for G12C, specific drugs that hit the mutants. There are Marathi and ourselves have shared some data, and I'll certainly share a little bit with you around preclinical G12D specific compounds. But there's a second class of compounds that we've also invested a lot of research into, and that is coming up with pan-KRAS drugs. So drugs that, if not all the major mutants, a number of the major mutants that they actually inhibit or even degrade, and I'll talk to you a little bit about what degradation is, simultaneously. And these would obviously be extremely powerful drugs if we're successful. Now, this year is fantastic, as we heard at the beginning. Now, this is just the beginning, but to have the fantastic work by the colleagues at Amgen and Marathi to come up with sotorasib and adagrasib in the clinic that works not only in non-small cell lung cancer, but as we've heard very recently at ESMO, in colorectal cancer. This is really a fantastic achievement. I'm sure Parsi will talk more about this in the afternoon, so I won't dwell on the data here, but this is really a fantastic start. At BI, we're really taking a long-term approach to drugging KRAS. We want to drug all the forms of KRAS, all those mutants, including wild-type amplification. And essentially, it's this volume of signaling we talked about. In cancer, the volume is really loud, if you like, and the job that our drugs have to do is to turn that volume back down to normal. And if we do that in cancer cells, which are essentially addicted to this loud KRAS volume, they will die. They go into what's called apoptosis, or programmed cell death. And of course, we need to kill as many, if not all of those tumor cells to get long-lasting effects. Importantly, to get those long-lasting effects, we think we're going to need to do two additional things, namely blocking feedback. I'll talk to you a little bit about that. It's sort of complicated, but hopefully I can explain that to you. And also, very important, we need to get ahead of resistance, and we're really trying to do all this in parallel so that we don't have to wait as long as we've had to wait, for example, in the EGFR field, to really get longer benefits. So this is an overview of what we've got and disclosed so far at BI. So we have an approved EGFR inhibitor, afatinib. We have two clinical compounds, a G12C inhibitor and a SOS1 inhibitor, 1709-1963 and BI 1823-911. But importantly today, and I'll share just a sort of a vignette on each, importantly preclinically still, so we still have to get these compounds through to the clinic, but we have G12D selective inhibitors, we have pan-KRAS inhibitors, and excitingly, we have pan-KRAS degraders, and I'll talk to you about that. I just don't want to confuse things too much, but this feedback, it's called negative feedback. It's very important, and it is a big challenge to turning the volume down in signaling. And essentially, this is the phospho-ERK, or ERK is at the bottom of this signaling, and that's when it's phosphorylated, it's on. And what signaling does is, while it's on, these feedback nodes are continually turning the signaling off. Right, now this sounds a little bit weird. You know, why, if you want to turn something on, would you simultaneously turn it off? And you can see with these red arrows that that signaling turns itself off at multiple points. And the reason it does that is to stay very robust. So as soon as we try to inhibit this signaling with drug molecules, it turns itself back on. And this is this signaling feedback that we need to get on top of if we're really going to turn the volume down and keep it down. And SOS1 is in particular a very important feedback node, and that's why I just want to highlight this feedback mechanism. Pasi will probably talk about this feedback, particularly in colorectal cancer, that comes through via the receptor tyrosine kinases, which I think explains why the combination with EGFR inhibitors, for example, in that space, gives a significant benefit. So for SOS1, we're the first, you know, we have a motto at BI, we want to be first for patients. So it'd be great for you to meet the team here in Vienna. So I really, if anyone's in Vienna, I invite you all. The team really here is motivated to really get molecules into the clinic for KRAS cancers to be first for you. We were successful with SOS1, and I call it the pacemaker. So if KRAS is the beating heart, just like our hearts have a pacemaker that in this case use electrical signals to help our heart beat, SOS actually, it's a chemical signal in this case, but SOS actually helps turn KRAS on. So without SOS around, KRAS can't beat actually. So it's actually twofold. One, it's the pacemaker, but it's also that feedback node. And you see here, you've probably seen these graphs. These are so-called xenograft models. So they're mouse models where we take a human cancer cell line, put it under the skin of a mouse, let it grow, and you see this black curve here. This is untreated and grows obviously a lot faster than a human tumor. But here, we look at untreated mice. This is an average of about eight mice here together. 8 together. You compare this growth to a treated group. So here we're treating the green one, I should say, is the SOS1 inhibitor. You see, you get good tumor control, but not really strong control. Trametinib is a MEK inhibitor, which does about the same. But when you do both, which is also blocking this feedback, you see very, very strong control. Even off treatment, the growth is only slow, and if you start treatment again, you get control of that again. And I'll show you some data also, some exciting data on KRAS G12C combination with SOS1 as well. this is exactly what we see now. If we do exactly the same thing, here again, xenograft model, now with G12C, in this case, it's our BI's G12C inhibitor. You see good in this is a lung cancer model with G12C. You see good control, basically the same picture. SOS1 gives moderate control. The G12C inhibitor gives also good control. But when you combine both of those, you see this very, very strong inhibition of the cancers. And this is why we're actually pursuing 3 clinical trials now with the sotorasib, adagrasib, and our own G12C inhibitor in combination with SOS1. So there's a lot of, this is across non-small cell lung cancer and colorectal. It will be very exciting to see the results of that. And I mentioned resistance at the beginning, and I really want to share, you're probably the first people to see this, or almost the first. I think Marco Hoffman presented this data just last week or a couple of days ago. So we're really trying to look at resistance. And in this case, this is again a xenograft model. Each curve now is a mouse, and we treated with MRTX849, so the Mirati G12C inhibitor. And you can see it gives wonderful control on a lot of tumors in mice, but there are tumors in some mice that escape treatment. So we actually took these mice here, this group here and this group here, and actually randomized those again, and then started a new experiment here. So all of these mice were essentially put into this point here. We continue to treat with the Mirati compound by itself, and as you would expect, because it was escaping here, it failed to control these tumors. If you take the data that we've just seen clinical data for, so this is Mirati plus the EGFR inhibitor, cetuximab, you see good control. But if you then take Mirati now and combine it with our SOS inhibitor, you see even stronger control of those tumors. So I think I just want to emphasize this, with regards to resistance and finding the right combination partners. Beyond G12C, of course, as well in the future, but certainly still for G12C is an important area of very active research. Now, I want to make sure there's enough time to answer all of your questions. So I've just got 1 or 2 slides left, but I really want to talk to you about what are called PROTACs, proteolysis targeting chimeras. It's another one of those terms, Terry, I apologize. But this is a really hot field now. The last decade, a little bit less. This is this completely new type of drug molecule has come not only to research, but also the first ones into the clinic. And the conceptual difference is our classical drug molecules are just like sort of like keys, and they turn these disease causing proteins off. What PROTACs actually do is they stamp them. And did you see that little video? I didn't see it. Did it work? It might have worked. Did it work? Yeah, there we go. It actually causes these proteins to get completely degraded, completely shredded, and the whole protein itself disappears. So this is for various reasons I won't go into today, but it really is a very exciting new class of molecules, and we've been for quite a while now particularly interested in trying to make degraders, so KRAS PROTACs, so we can actually not just turn KRAS volume down, but actually shred the whole protein. And we've been successful so far in the preclinical space to actually discover these, and to my surprise, actually, these KRAS PROTACs, they actually degrade the majority of the main KRAS mutants. So they're actually pan degraders. And you can see here a list of all the mutants. And this number here is again a concentration at which 50% of KRAS is degraded. So you can see, this is 1 nanomole in G12D, and 50% of KRAS is degraded. And you can see it's a number, only a number, G12R, Q61R, Q61K, and L, which are mutants that have KRAS essentially stuck in the on conformation, where these drugs work less effectively. But for all the other mutants, where there's a significant amount of KRAS in the off conformation, these PROTACs are extremely potent. So that was I appreciate it's a bit of a sort of whirlwind overview, but I really think we're on the cusp now. If we can really continue on the path that we're on, I think we're on the cusp of getting a number of new medicines in the KRAS field, and it's just a reminder of the drugs that molecules I've talked about. And one last little aspect, I sort of via Twitter and various other media, patients have been sort of asked about, what clinical trials are available. This is just an offer, Terry. We can work this out sort of offline, but Waltraud Pasteiner, she's a wonderful researcher in our team. She's offered to provide a list. I think there's about 200 at the moment of clinical trials that recruit patients with cancers with KRAS mutations, and we can provide updates to that to really provide a resource for the kickers. And with that, I must show you the face of the team. It's not me, it's these people here. The photo at the top here is an old one, a few years from. We have a Summerfest here in research every year. This is actually very recent. We were up, in outdoors to make sure we didn't have any infections. But yeah, fantastic team of researchers. I'm humbled to be part of the team, and yeah, hope we can continue to contribute to all of you and come up with medicines. Thank you.
Thanks so much, Darryl. I really appreciate your coming and joining us today. We do have a couple questions for you, if we could.
Yeah, I'll just stop screening. Stop sharing.
No, we can share the screen. That's okay. I'll share.
Right.
Yeah, stop hearing others sharing. But first of all, I got to tell you that hearing your what did I didn't catch the name. Was it Waltraud?
Waltraud, yeah.
Thank you so much for making that offer. This is one of the biggest problems, the biggest challenges that we have as patients, being able to find the right trial and being able to identify, even just have a starting point does not involve the .gov website nightmare. So thank you. That is going to be here. So totally down with it. Just like keep it up. Okay, so that's first. And then we have one question. I get a couple questions coming in online here. So the first question was, this is from Jessica. What's the tolerability of the sauce one so far? The sauce
we haven't disclosed anything clinically, but I can say that preclinically, it's always been very well tolerated. Okay, I'll take it. But thus far, we haven't reported any data on from the clinical trials, so I'm not in a position to share that. Okay, well, no, that's okay. That's okay. Obviously, you're still looking into it, so absolutely there is a possibility that something may actually be working well. So if it wasn't going to work well, then we probably wouldn't keep looking. By the way, if you guys have orange cards on your tables, if you have questions, just go ahead and write them up and shoot them up with the runner. Okay, I saw one more question come in, so pardon my technology here. No problems, no problems. Okay, and then Lisa wants to know, probably what a lot of us want to know, what is the future for the 12D, 12V as far as like timeline? Can you give us, I know I saw on the slide, there was a do not post. And so so y'all know, if you're not used to doing these researchy sort of science stuff, I didn't know this. You'll see things when you attend that say don't post and really don't do that. So they're just telling you something and you won't be seeing that later. So do you have any sort of like enlightenment you might have for us on the 12D and 12E? Yeah, I thought you were going to ask this one. I was jogging this morning thinking, how am I going to give you a proper answer? So one thing that's important to me personally, but also to certainly to BI, we don't like to sort of predict the future. I'm still of the opinion no one can do that. I certainly can't. But let me just give you a little bit of a perspective. So what we've done, we're really trying to share the latest data. So that's sort of hot off the press. Wanted to share with you today. If you take it takes us about 6 years to get from a start of a drug discovery project in oncology at BI to get to first patient in, treating the first patients. That's 6 years. The last 12 to 18 months are a really important phase where before that we have a candidate, but we really need to then answer the question, is it safe enough? There's quite a few other questions in that phase as well, but that's one of the biggies, and where we have to do toxicology studies, and anything can happen in that phase. We've had molecules that just sort of sail through and look wonderful, but we've had to sometimes go through this cycle three or four times until we get a molecule that's actually safe enough to go into clinical trials. Okay. And you might hear this sort of IND filing, IND enabling phase. That point when you get to actually file for an IND, this is the sort of the name for the international name for the drug. When a company actually gets that IND filing approved, they've got through all that toxicology and it's safe. That's sort of and I don't like to say, we are predicting IND filing in Q2 something. I don't think it's responsible because, it's usually based on the optimism that the molecule is going to be fine. But so, that might give you a little bit, you can start to do your own maths and things, and of course, for every one of our KRAS projects, I'm hoping that, we have luck through these toxicology phases and we don't have to go back 12 months and try again. So that gives you perhaps a bit of a feel for the time frame we're talking about.
Okay, I'll take it. So but it's good to know that you guys are working on it. And I got to say that BI particularly, you went out of your way to mention both Mirati and Amgen, and all three of y'all are top tier sponsors here of this event. And so I appreciate that. But we also appreciate as patients to know that the companies are working together in order to come up with treatments for us. Because we really, when it comes down to it, we just want to have the drug that works. We want to have what works for us and whatever that looks like. And we're sorry for all the loss of life with mice and all the other things that go on behind it. But 6 years is a long time. And if you start looking at the research numbers, I mean, those things start dropping off at 5 years. And so we need to start speeding it up. So we appreciate everything you guys are doing. Does anybody have any questions, just jumping off, just burning issues here? I'm not seeing any burning issues here.
Terry, can I just say something just one last comment on the timelines? They're average timelines. Now, there's no such a thing as an average project in drug discovery, and there is certainly no such a thing as an average patient, right? So all these, medium PFS and, all these are all average numbers, and you can't go back the other way, right? To an individual patient or to an individual project, that prediction doesn't work the other way. Every one of us is unique, and each project is unique. So we certainly here in Vienna, we don't believe in averages. We believe in outliers. And I think also the speed that Amgen, for example, and Mirati are also very fast, brought G12C compounds to patients and to the market. How fast the mRNA vaccines, for example, have been shows that, if we think big and we're courageous, we can be outliers rather than average in terms of speed. So I just give you the averages a little bit to give a feel, but that's not our mindset for sure.
Oh, I totally down with thinking big and being courageous, because that's just really what it takes. So, okay, so let me ask you a question, since I don't see anything coming in from the audience here. Okay, so could you just get a little bit more clarification as far as there's wild KRAS in our bodies just running crazy and free. And is that everybody? I mean, do I read that right?
Yeah. So KRAS is wild type KRAS, so which means sort of the normal KRAS we've got in our bodies. It's not evil. It's good. We need it for our cells, dividing cells in particular. But you saw that sort of off and on thing. It's just at a very low level normally, right? So that's the sort of normal function. When it gets mutated, it becomes really overactive, and that's what causes the problem. So that's sort of the difference. What we don't know still to this day, once we get once we get the first pan-KRAS molecules into the clinic, then we'll find out what's the consequence of inhibiting or degrading wild-type KRAS. So that's still an open question. In EGFR, for example, we know if you inhibit EGFR wild-type and not the mutant, so the normal EGFR, you get a lot of rash and diarrhea. We know that for KRAS, it's simply unknown. So that's one of the reasons we want to continue until we know the answer to that question, pursue both the selective and pan. My hope, of course, is that there won't be any significant toxicity, because that will mean that we can really find a utility for these pan drugs.
Okay, so when you're going and looking at this, like say the doctor takes a pathology, is doing a biopsy, I mean, can he tell the difference between the wild KRAS and the non-wild KRAS?
Yes. So that gene, the chromosome and then the gene, when you get the genetic tests, it's actually that gene, those 46,000 base pairs, letters, make a big long word, 46,000 letter word essentially. That's what's measured in these genomic tests. And then they look at that one position, in position 12 or 13, that is different. It's simply a spelling mistake. And these genetic tests see the spelling mistake very clearly. And if there's a spelling mistake there, it's mutated. If there's not, it's wild type. Okay,
and so basically you need an app to fix our misspellings.
Yes. This is, cancer is a disease of our genes, right? And actually, as our cells divide, spelling mistakes are being made all the time. But we've got all this machinery to fix, sort of rubbers, fix the spelling mistakes. Or if the spelling mistake's really bad, the cells get actually discarded deliberately. And cancer is actually sort of over, apart from childhood leukemias, but it's actually the accumulation of about, let's say, a half a dozen spelling mistakes in different proteins. KRAS is a very important one, but they add up together to form cancer. So it's sort of, that's why it takes 30, 40 years for this accumulation to happen. So it's really bad luck, right? It's this combination of the wrong spelling mistakes over time. Yeah.
So it's kind of a spelling and multiplication error both combined. Yes. Oh, okay, fantastic. Okay, and then can you tell the difference between a mutation and amplification?
Yes. That's also very clear.
Okay. All right.
Yeah, the same genetic tests can do that. It's certainly doable and being done. I think for the wild type amplified, it's been a bit, I think, a little bit forgotten. And there may be some tweaking of some of the biomarker assays that need to be done for the wild type amplified. But this is something we scientists will be able to manage.
Right. And then, okay, so I'm just going to hit you up with a couple like just random questions that I get all the time. Okay. So pan means, when it says pan KRAS, that means panel, right? Is this like a panel? So it's attacking more than one.
Essentially a panel, yeah.
Panel, right?
Yeah. A spectrum.
It means make you wiggle in your chair, Darryl. I'm sorry.
Sorry. That's the first, every time I get the first question for the first time, I must wiggle.
Okay, great. All
I'm
right.
nervous about the next one now.
These are the sorts of science questions we get here.
No, look, I really appreciate it because, you know, we're talking all this jargon of science. I know that, you know, and I try to, you know, that's why I try and draw pictures and use proper language in communication. But, you know, I still realize, you know, interacting with yourself and all the wonderful people here that
you're all good. You're all good. You're all good. Okay. And so when we hear like, this is a pathway treatment or pathway drug, what does that mean?
Yeah, so the pathway, that sort of wiring diagram I drew, that's what we call a pathway. So it's a sort of, it starts from outside the cell, starts with a certain protein, and then works its way down to the nucleus. In reality, there's a sort of network of all these pathways, but it's essentially sort of molecules, proteins in the cell connecting together to form a signal, just as an electric sort of circuit.
Okay. All right. Now I've had this question. I don't know the answer. A lot of times I'll ask you a question I know the answer to. I don't know the answer to this one, so be afraid. Okay. I would like to know, how big is this, that like if I know you're looking at something microscopically. Okay. But how big, give me some point of reference to how big this really is.
KRAS.
Yeah. Like, the original, like a mutation, like when you're looking at it, how big, how small is this little critter that started this problem for me?
Yeah. Really, really small. So you can't see it with your eye. So a hair, the thickness, I haven't got any, unfortunately. One of your hairs, that's about the smallest that a human eye can actually see, right? And I think it's about, what is it, about 10 microns or something like that. I'm guessing a bit here. I'd have to probably look it up. Actual KRAS protein, it's probably, I don't know, it'd be hundreds of millions of times smaller than that.
Okay.
So really, it's actually, and even as far as proteins go, it's one of the smaller ones. So it's really, really small. That picture you saw, that we have to use X-rays. So we actually have machines here with big X-rays. We make a tiny little crystal that you can just see with your eye, and we freeze it down to almost absolute zero, and we shoot X-rays at it to be able to detect those atoms and then reconstruct the picture. So it's you know, it's really, you need the most powerful techniques we've got to be able to see it. So
on like one hair, there'd be like 100 million or something?
In a cell, how many KRAS molecules in a cell? That's a good one. I better look that up before. I'm going to tweet that tonight.
Okay? All right, you tweet that. Okay, I'm sorry. I did not mean. I thought like I'd be able to get like a simple answer, but
yeah, I know.
Sorry, I did not mean to put you under that pressure.
I know the concentration, but the actual number of molecules I'd have to work out.
Yeah, well, you know, so okay, and all right. I don't know if you're going to be allowed to do this or not, but can you can you give us a Kick Cancers KRAS?
Absolutely.
All right, let's do it. You want to do it together? Or you want to do it?
Yes, please. Yeah.
All right. Are you going to do it with the room?
Oh,
all right. Awesome. Room want to do it with us? Okay. All right, let's do it with the room. Okay. Okay, can everybody see me? I'm going to do a count. I'm going to do 1, 2, 3, and then we'll Kick Cancers KRAS. Okay. 1, 2, 3. Kick Cancers KRAS. All right. Thank you, guys. Thank you so much for joining us this morning. And thank you guys for everything at Boehringer-Ingelheim that you guys are doing. Eventually, by the end of the day, maybe I won't stutter over it. And thank everybody there for us. And you guys keep working to do what you got to do to Kick Cancers KRAS. And we will see you on Twitter. Thank you.
Absolutely. So much. Yep. Don't worry. We're not going to give up anytime soon.